Welcome in this presentation about the biology of the SARS CoV-2. Viruses can be of two types depending on their outer membrane. The first one are the naked viruses. These are the most simple form of viruses which consist of only two things. Number one, an outer membrane or capsid made up of protein, and number two, a viral DNA or RNA, the genetic material within the capsid. The common examples are polioviruses, rotaviruses, adenoviruses, etc. The second type are the enveloped viruses. These enveloped viruses have an additional membrane called as envelope outside of their membrane of the capsid. This envelope is mostly made up of lipids derived from the host cell membrane. From the envelope, some viral glycoproteins remain projected and play a crucial role of the adherence of the virus to the host cell. Some common examples are chickenpox viruses, herpes viruses, measles viruses, etc. The enveloped viruses are less stable than the naked viruses because their envelope is more susceptible to the external factors like temperature, chemicals, etc. But on the other hand, due to the flexibility of their envelope, they can more aptly adapt with the host immune system. That is, they are more adaptive than the naked viruses. Now we have to know about a definition or a particular term called as virion. What are the virion? Virions can be defined as the smallest invasive unit of viruses. To invade into a host cell, a virus minimally needs two things: the capsid for penetration and the DNA or the RNA for the infection. So this capsid and the DNA or RNA together form the minimal unit of virus called as the virion. The severe acute respiratory syndrome coronavirus 2 or SARS-CoV-2 belong to the Coronaviridae family of viruses, and within the beta coronavirus, the newly evolved SARS-CoV-2 is actually a mutated strain of the original genus SARS-CoV. The coronaviruses are named so due to their resemblance with the solar corona under electron microscope. The projecting glycoproteins or the spikes resemble them with the solar corona structure. Previously, there were 6 known coronavirus species which can cause illness to human beings. Now, including this newly evolved SARS-CoV-2, the number has reached to 7. But out of the 7 coronavirus infect which can affect human beings, only 3 can be lethal. Number 1, the newly evolved SARS-CoV-2. Number 2, the original SARS-CoV. And number 3, the MERS-CoV. Features. Under electron microscope, the SARS-CoV-2 appear spherical to elliptical with an average diameter ranging from 60 to 140 nanometer. SARS-CoV-2 or the coronaviruses are a type of single-stranded positive stranded RNA virus. Positive stranded RNA means the RNA that the viruses have can readily act as a mRNA and need not to be replicated to a same strand like other RNA viruses. In a normal cell, the genetic code remains embedded into a DNA molecule. From this DNA, the genetic code is first transcribed to an mRNA or messenger RNA. Then that messenger RNA is finally translated to protein. The RNA that we find in the coronaviruses can directly act as a mRNA or the messenger RNA, and thus they take less time to affect the host cell. The viral genome of the SARS-CoV-2 contains about 10 open reading frames or ORFs or 10 translational units. The first two-thirds of the viral RNA code for two large polyproteins, PP1A or polyprotein 1A and PP1AB or polyprotein 1AB, which are then processed to 16 non-structural proteins, which ultimately contribute to their replicase transcriptase complex. This replicase transcriptase complex is employed in the viral replication or translation process. The next or the last one-third of the viral genome code for four primary structural proteins: S for the spike protein, E for the envelope protein, N for the nucleocapsid protein, and M for the membrane protein. The spike protein is very important for the adherence of the virus to the host cell. The spike of the SARS-CoV-2 is made up of two subunits, S1 and S2, with a cleavage site in between, and the extended end or the outer end of the spike act as the receptor binding domain or RBD. To invade into a host cell, this RBD should be received or recognized by some receptor on the host cell membrane. The particular type of receptor that the SARS-CoV-2 require is the angiotensin converting enzyme 2 or ACE2 type of receptor. This RBD of the SARS-CoV-2 are first received by the ACE2 on the host cell. Then a protease released from the host cell will break the cleavage site and will expose a fusion sequence from the virus, which will enable the virus to fuse with the host cell membrane. Patients suffering from the cardiovascular diseases or kidney problem found to have more SE2 receptor than others on their cell. That is why they are at more risk or increased risk under this SARS-CoV-2. The SARS-CoV-2 or the coronavirus 2 were primarily named as the novel coronavirus. The word novel implies an unique feature of the virus. The particular cleavage sequence that they have in their spike is unlike their counterpart. Unlike the other coronaviruses, the SARS-CoV-2 has a polybasic cleavage site which is cleaved by a furin protease type of enzyme. And these furin protease are very commonly present on the epithelial tissue of the lungs and the respiratory system. That is why they can cause severe problem. Try to answer these questions. Thank you.